Hello, everybody. Welcome to today's video lecture, and we continue covering muscle tissue. In this lecture, we will look at physiology of muscles, right? So let's go ahead and begin. So we have 4 phases of muscle contraction, and they are activation or excitation. Then let's just look at 3 and 4 after 1, right? So we have excitation, contraction, and relaxation. That makes sense, right? So you need to activate or excite your skeletal muscles. Otherwise, they cannot contract. So you need this electrical signal from nervous system to activate your skeletal muscles. Then they will contract and then they will relax. And there is one more step that connects excitation and contraction, and it's called excitation contraction coupling, right? So that's why I wanted to cover step three before I talk about step 2, right? So again, what do we have? What are our 4 phases? Excitation, contraction, coupling, contraction, and relaxation. Now, activation is neural stimulation at a neuromuscular junction. We will look into detail of this activation process. Excitation, contraction, coupling is generation and propagation of an action potential along sarcolemma. And final trigger is a brief rise in intracellular calcium level. And then muscles, sarcomeres will shorten, muscle will contract, and then muscle is relaxed, right? Okay, so we need this activation or excitation, and it happens at neuromuscular junction. So where this junction is, right? So neuromuscular junction is a site where motor neuron meets muscle fibers. Muscle's first response is here. And excitation by neuron is the only functional way to activate contraction. So if you look at diagram over here, so you see a neuron, and this is a special type of neuron. It's called motor neuron. And there is area where this motor neuron meets a muscle fiber. So here's the muscle fiber. So here's this area magnified where they actually meet. So you can see this structure that is axon. It's an axon terminal. So that's a part of neuron right here. And this is your muscle. And you have like this wavy line, right? This line that I'm drawing is actually sarcolemma. Sarcolemma is the membrane of muscle cell. And this area is called motor. It's right here, motor end plate. So motor end plate is a part of the sarcolemma where we have synapse between axon of motor neuron and a muscle cell, right? So again, we have motor neuron, it meets muscle fibers, right? In the area where they meet, we call it neuromuscular junction, right? So we have axon terminal, that's a part of axon, and we have motor end plate, that is part of muscle cell, right? And this is where we have activation or excitation of our muscle cells, right? Okay, now let's get to the next slide. So what exactly happening? So here we have our axon terminal, and here we have our motor end plate of the muscle cell. Now maybe we're gonna do it here. Yeah, I think we will get back to this I'll go back to this picture. Now inside, and I remove my drawings. So inside this axon terminal, we have a vesicle. So here is nice magnified picture over here. So here we have a vesicle. And inside vesicle, we have special chemical. It's a neurotransmitter, and it's specific neurotransmitter called acetylcholine. That's abbreviation, ACH. Now, this chemicals was pre-synthesized before, and it just stored inside those vesicles. Now, when you want to contract your muscles, your neurons, you see shown here in red, gonna generate electrical impulse, right? So that's electricity moving along your neuron. And when this electrical impulse reaches the terminal, it activates those vesicles. And there is a whole mechanism how those vesicles are activated. We are not going into such details. We're just gonna say that when electrical impulse that is called action potential reaches axon terminal, and we already have this vesicle with acetylcholine, but now those vesicles start moving toward the synaptic cleft, toward the end of the terminal, and they're gonna fuse because they have phospholipid membrane and this axon terminal covered by phospholipid membrane. So vesicles gonna fuse with neurolima and release acetylcholine in this synaptic, inside the synaptic cleft, right? And now acetylcholine will attach to its receptor. Where do we have a receptor? On a muscle cell, on actually on the sarcomere, right? On the membrane of muscle cells at a motor and plate. So where we have this wavy structure, right? We have this motor and plate. We have receptors for acetylcholine. Now you can see acetylcholine binds to its receptor. And when this happens, this allows sodium, right, to move in. So here's our sodium moves in. I don't know, do you see this plus of sodium? Because sodium has plus. I don't have my glasses, but so sodium start moving in, right? So we have movement of sodium inside a muscle cell, right? This is our excitation. We actually, you know, it's a part of excitation, we exciting muscle cells, because what we need to do, we have electrical impulse over here. Now, when electrical impulse reaches this synaptic cleft, it cannot move through this space, right? And that's why instead of electrical signal, now we're moving chemical signal, acetylcholine. But when acetylcholine attaches to its receptor, and when it allows sodium to move in, so you can see sodium moves in, now we can generate electrical signal in the muscle cell. So you see these arrows here, the white arrows. So now we have action potential traveling along sarcomere of muscle cell. So really the point of this neurotransmitter is how to, so imagine that you have wire at home, right? And you have a cut in your wire. If you have cut, electricity cannot move through this area that, you know, that the space, it has fluid here, space, electrical signal not gonna move from axon to muscle. And we want it to move from axon to muscle. That's why we release this chemical, acetylcholine. Acetylcholine causes sodium to move in. And when sodium start moving in, it will open other channel, sodium voltage gated channel. And we have lots of sodium moving in, right? And when charged particles move in and out, that's what electricity is. So now we generate action potential that travel through our muscle cell membrane or sarcolemma. So we go from one action potential of neuron to another action potential of muscle cell. So that's our electrical impulse. And this is our excitation. So we made this muscle cell excited because now electricity moving along its membrane. That's excitement, right? So now this action potential move along sarcolemma and it moves through T-tubule. So that's our T-tubule. And in a T-tubule, we have a special proteins called Dhp receptor. And when electrical signal moves through this receptor, it activates the receptor. Dhp receptor activates Rye receptor. And Rye receptor are actually gated channel. So you see it's closed over here. And it's closed, so it's keep calcium. That's a calcium. Oops, that's a calcium. This blue part shown here in blue part. So when Rye receptor is closed, calcium is inside sarcoplasmic reticulum. But now when all this excitation happens, we stimulate Rye receptor, it opens its gate and calcium enters intracellular fluid. So we have lots of calcium now inside this cell. And this is called excitation contraction coupling. Because let's go back over here for a second. So we need activation, right? We had neural stimulation at a neuromuscular junction. Then we have generation and propagation of action potential along sarcolemma. So we move from one electrical signal that is traveling through neuron, right? It reaches this gap, synapse. Acetylcholine is released, neurotransmitter, and it causes action potential now to be generated along sarcolemma. And finally, when they reach those proteins, right, it allows calcium to enter intracellular space, and calcium was stored inside sarcoplasmic reticulum. Now we have calcium inside cytoplasm or cytosol of a cell, right? So here we good. Now we have calcium. So we're done with activation and excitation contraction coupling when we release this calcium. That was our goal. So actually, the whole reason, let me again remove my drawings. The whole reason for this process was we want calcium inside cytoplasm or sarcoplasm of this muscle cells. We need this calcium, right? So far it makes sense, right? Why do we need this calcium? We're gonna discuss next. Okay, so now why do we need this calcium? So just before we get to this point, this slide covers pretty much what we already discussed, right? So that's our excitation, right? Release of acetylcholine, then traveling of action potential through T-tubules and release of calcium over here, excitation, contraction, coupling, right? So that's we discussed already. The only thing shows over here when after acetylcholine is released, the extra amount of this chemical need to be removed from the synaptic cleft, and we have a special enzyme, acetylcholine esterase, that will degrade extra amount of acetylcholine over here. Right, and when we don't have acetylcholine anymore, we don't have excitation, and calcium returns back inside the sarcoplasmic reticulum, and then muscles can relax. Right, so remember, when we have calcium present, muscles gonna contract. When calcium is inside sarcoplasmic reticulum, muscles relax. Right, this is what this diagram shows. Okay, now let me let me show you this first. Now, imagine your muscle, right? So for example, take your, well-known bicep brachii. So if you have your muscle, right? So for example, that's a whole muscle, right? So have a tendon. And muscle is made of individual muscle cells. So here, if you take one muscle cell, right? Now this muscle cell is divided into units called sarcomeres, right? So here we have our sarcomeres. So repeating units that are sarcomeres. Now imagine what happened if each sarcomere became shorter, right? So now look, this is how long my sarcomere is, right? How about, let me draw a little bit better for us. Okay, so anyway, so here's our muscle, right? So here's the muscle. And then we're gonna look at individual muscle cell. And let's say I have, I don't know, 1, 2, 3, 4 only sarcomeres shown, right? So how long my sarcomere is? So my sarcomere is that long. Now, what if each of these sarcomeres gets shorter? So this sarcomere now becomes this short. This sarcomere became short. And this one short. And this one shortens, right? So now, okay. So 1, 2, 3, 4, 1, 2, 3, 4, right? So I have the same 4 units, 4 sarcomeres here, 4 sarcomeres here, but now my every sarcomere that I have is shorter, right? So now if my sarcomere is shorter, what happened with my entire muscle? My entire muscle is shorter, right? So that's how your muscles contract. So now when we have this calcium present inside cytosol or cytosol of muscle cell, right? Oh, it's sarcosol. No, cytosol, okay. So we have calcium present inside the muscle cell. It's released from sarcoplasmic reticulum. Now we can make sarcomeres shorter. And if every sarcomere shortens, the whole muscle cell gonna be shorter, the whole muscle will contract. Right now, how to shorten the sarcomere? So if you remember, over here, you can see structure of sarcomere. We have M line in the middle. We have 2 Z discs or Z lines. From Z line to Z line, we have 1 sarcomere. Now, what attached to M line is myosin. So this red molecule here is myosin. What attached to Z line is this blue molecules called actin. Now, myosin, if myosin heads connect to actin and they pull this actin towards the midline. So imagine that those are little arms of myosin. If myosin can grab on actin and it can pull actin towards the midline, look, it's happened right here. See how my myosin here is not touching M line, right? There is a distance. But now if myosin can pull actin towards the midline, then the whole sarcomere gets shorter, right? So, and this is our goal because we want to make each sarcomere shorter. So now we need myosin to grab on actin and pull actin towards middle, and then the whole sarcomere shortens, right? So let's see how this is done. So this is your actin, right? Now actin has these units that show here, this nice round structures. And this was shown in a dark spot here. This is myosin binding site, right? So here's a myosin binding site. So for myosin to grab on actin, this side need to be exposed. But actually, when muscle is relaxed, look, we have another protein here covering those binding sites. And this is called tropomyosin. So you see how tropomyosin covers it? So myosin cannot grab on actin. Now, when we have calcium, calcium attaches to another molecule called troponin. And when calcium attaches to this troponin molecule or this troponin molecule, it causes conformational change and tropomyosin shifts away from binding sites. And now we have binding sites exposed. That's why we need calcium. Remember when we were going through all this process over here to release calcium from sarcoplasmic reticulum. Well, now we know what we need this calcium for, because calcium can bind to troponin, and it will cause tropomyosin to shift. Those molecules, they shift away from myosin binding site. And now we're good. Now myosin can bind on actin. And here we have the whole process explained quickly in pictures, how this happened. Right, so myosin, let's actually, I want to start with number 4 over here. So this is our myosin. Myosin has 2 heads, and it has a special site over here where ATP binds, right? So here ATP binds to myosin, right? And what myosin does, it splits ATP into ADP and phosphate group. So it removes one phosphate group from ADP. And when this happens, the myosin, so it says here, myosin heads split ATP, and they become reoriented and energized. So now those heads are energized. We call it cocking position. So they now in a state when they have enough energy to grab finally this actin, right? And other things need to happen. Actually, the myosin need to let phosphate group go. So it loses phosphate. And when it loses phosphate, it connects to actin. So we call it cross bridge. So formation of cross bridge. So again, myosin has ATP attached, right? Myosin splits ATP into ADP, adenosine diphosphate, and phosphate group, and it became energized. Now, when it loses phosphate, it grabs on actin, right? Then, and we call it cross bridge formation. Then when it loses ADP, it pushes actin towards the M line, and that's what we need. So myosin heads rotate towards the center of the sarcomere, and it kind of pushes, push or pull. Yeah, I think it's pull actin towards the midline, right? Pull the actin towards the midline, and this is called power stroke, right? And now it stay attached, but it doesn't have ADP or phosphate group or ATP. Now ATP molecule attaches to myosin, and it's allowed to detach myosin from actin. So as myosin head binds to ATP, the cross bridge detach from actin, and we start this process again. As long as we have calcium to move this tropomyosin and expose this myosin binding side that you see here in black, right? And as long as we have ATP. Right, so let's repeat one more time. So making sense of all this craziness over here. So we have excitation, we have electrical signal traveling along neuron, reaches axon terminal, causes vesicles, synaptic vesicles to fuse with neurolema, with membrane of neuron, release acetylcholine into synaptic cleft, right? Now, when acetylcholine is released, it binds to receptor, acetylcholine receptor. This causes sodium moving inside the cell, and this will generate action potential or electrical signal along sarcolemma, right? This is our excitation. We excited the cell. Now, cell is so excited that electricity is moving along its membrane, and it moves along T-tubule. It activates Dhp receptor. This receptor activates Rye receptor. Rye receptor open a gate, and calcium flow from a sarcoplasmic reticulum to the sarcoplasm. Now we have calcium. Now when we have calcium, calcium attaches to troponin and move away tropomyosin. Now when tropomyosin moved away from myosin binding sites, right, we can have myosin. Myosin will form cross bridges with actin, so we can have myosin grab on actin, right? So here calcium is present. It's attached to troponin. Tropomyosin is out of our way. Now myosin, because it has ATP attached, it splits ATP and it's became energized. Then it loses phosphate and it forms cross bridge with actin. Then it loses ADP and it go into power stroke. So it pulls actin towards the midline. Then new ATP attaches and we detach cross bridge. So we can start all over. And now when we're doing that, right, we actually pulling actin towards the midline, right? And sarcomere now shorter. So here's our sarcomere when muscle is contracted, and this is our sarcomere longer when muscle is relaxed, right? So that pretty much what's happening, and this is called sliding filament theory or sliding filament model that, guys get Nobel Prize to discover this model of contraction. And this is how muscles, sarcomeres get shorter and muscles contract, right? So let's just read this one. Thin filaments are pulled, a slide past thick filament, Z lines move together. So Z lines move together. I band becomes smaller, right? So that's our I bands where we have only actin. A band gonna stay the same because A band equals the length of the I don't know, I don't forget. Yeah, A band equals the length of myosin. So the length of myosin is not gonna change, right? So here's our A band. So you can see A band is from here to here. So I band became smaller, H zone shrinks, only A band stays the same. But the point is distance between two Z lines is getting smaller and muscle contracts, right? Okay, so actually I'm gonna stop right here because it's a lot of material to process, and I just better record one more video to cover the rest of this PowerPoint, right? So thank you for watching, and I hope it was helpful.